Now we are going to discuss about type 2 hypersensitivity. Type 2 hypersensitivity, which was described by Gel and Coombe. Okay, see, think there is an RBC, and due to some genetic susceptibility or due to some foreign particles like drugs, environmental factors. See, first of all, see, in our body there is RBC, right? Why the immunity is not attacking this RBC? Because this is understood by the immunity as self. Why self? Due to some proteins in the membrane, due to some receptors in the membrane, the immunity thinks that this RBC has self, so it won't destroy it. But due to some genetic susceptibility, the membrane of the RBC had changed, and some molecules which are antigens which came from the environment are changed due to genetic susceptibility. What happens? This immunity thinks that it is not self, and then produces, sorry, not RBC here, immunity produces IgG and IgM antibodies. What this IgG and IgM antibodies do? They go near the RBC and attached with the FC region. Okay, Ig got attached here, IgG antibody. What happened? The RBC is coated with or mutate, or due to genetic susceptibility the membrane had changed, and the immunity detected it as non-self, and it produced IgG and IgM antibodies. The IgG antibody had attached with the FC region in the RBC membrane. What happens then? Then ultimately the RBC should be killed, right? So the process by which this is killed has three types. Total three types. See, okay, total three types. I will explain this. In the blood, normally in the blood, there are some random proteins. What these random proteins do? These random proteins, whenever there is a bacteria, they will get activated or get activated by the immunity. They activate or they get activated by the immunity and help in the destroying of the cell by the immunity. There are random proteins, they help the immunity in destroying the foreign organisms like bacteria, viruses, anything, any foreign body. Okay, what happens? Here there is FC region, right? This FC region is detected by one of these random proteins. Okay, one of these random proteins. There is a name for this random proteins known as complement proteins. Okay, this is complement system, and the proteins are complement proteins. One of the complement proteins, C1, recognizes the FC region and it is coated with antibody, so we should kill it. So the complement pathway activates. Okay, why the complement pathway is getting activated? Because the RBC are coated with the IgG, and so if it is coated with IgG, it should be killed, right? So that's why the complement system is activated. In the first complement cascade, the first C1 is activated by recognition of FC region. So after C1 is formed, C4 is formed, then C2 is formed, then C3 is formed. Here there is an enzyme known as C3 convertase. This C3 convertase convert this C3 into C3A and C3B. That splits C3 and forms C3B and C3A. And then C5 is also formed. Then it is also splitted and form C5A and C5B. And then C6, C7, C8 and C9. These are also formed. Okay, now what we are discussing? We are discussing the RBC should be killed and there are three mechanisms by which it get killed. So first mechanism is, here there is C3 and C3b, right? So what this C3b do is, this C3b will go here near the RBC and it coats it attaches with the membrane. After getting attached, the macrophages recognize this C3b and engulf it and destroy. What happened? C3b coated this. Okay, here RBC is there. This not only this what RBC, it might be bacteria, it might be virus. Okay, it goes and coats the specific organism or any cell. After it is coated, the macrophage detect and kill it. Okay, easy, right? So what is this process of coating is called? The process of coating and foreign particle to get destroyed or engulfed by the macrophage or the immunity is called opsonization. And the particle which is helping in opsonization is known as opsonin. Okay, easy, right? So this is the first mechanism. That means C3b is going and attaching with the particle, and then opsonation is done. So macrophages detect and engulf it. So first mechanism of destroying is formed. Known. We know the first mechanism how it is destroyed. Okay, once again I will write the cascade here. Complement cascade. What? First C1, C4, C2, C3, C3a, C3b, then C5, okay, C5a, C5b, okay, and then C6, C7, C8, C9. This C5b initiates one more mechanism by adding with this all. That means this C5b plus, here what are there? C6, C7 plus C8 plus C9. These all proteins come together and form a complex known as MAC, that is membrane attack complex. Okay, this membrane attack complex goes near the particle to be destroyed, cell to be destroyed, and forms holes or pores. This MAC forms pores, and after the pores are formed, the water goes inside, the substances go inside from the external, and the cell gets swell and burst. Okay, this is the second pathway. Okay, then the third one is, see, if it is coated, should the cell finally it should die, or any other outcome also there? See, if this is coated with antibodies, should this cell compulsorily die, or is there any other mechanism can be happened? So it is not compulsory that it should die. See, for example, in myasthenia gravis, it is not compulsory that if in type 2 hypersensitivity surely the cells should die. It's not like that. See, in myasthenia gravis, what happened? Normally, acetylcholine, this is the acetylcholine receptor. Acetylcholine comes and attaches here. So, normal contraction of the muscle. But in myasthenia gravis, these antibodies, what happens? These antibodies come and attach with this. Why? This due to genetic susceptibility in the myasthenia gravis, the receptors are thought like non-self. So antibodies are produced against this. These antibodies come and attach here. After attaching to the receptor, what happens? This acetylcholine cannot bind. What happens if this acetylcholine cannot bind? What happens? There is no contraction and relaxation. So there will be contraction and relaxation disorder in myasthenia gravis. Okay, this is the one more outcome. We had known 3 outcomes. Okay, one more we'll know. One more outcome. See, this is myasthenia gravis, and in Graves' disease of thyroid, what happened? The thyroid stimulating hormone stimulate the thyroid and produce thyroid hormone. Okay, thyroid hormone is produced. The antibody in Graves' disease is produced as to act like TSH due to genetic susceptibility, as the antibodies are produced like TSH, so they will stimulate the thyroid gland. So this immunity produces antibodies which stimulate the thyroid and produce excess thyroid hormone. Understood? This is what happens in Graves' disease. This is the mechanism how immunity produces the antibodies like TSH which stimulate the thyroid, okay, and produce excess thyroid hormone. Okay, this is what happens. It is acting like TSH, right? So see, in the Graves' disease and in the myasthenia gravis, okay, myasthenia gravis and Graves' disease, did the cell die? No, the cell did not die, but the function is altered. Okay, the cell is not sure to die. The function may also be altered. Here there is one example of an important disease, that is good posture syndrome. Good posture syndrome. Okay, what happens in this good posture syndrome? See, in this good posture syndrome, 2 are affected. One is lungs. Lungs are affected, and one more, kidneys are affected. Okay, in the type 2 hypersensitivity, we are discussing about a syndrome known as good posture syndrome, in which lungs and kidneys are affected. How? See, in the lungs there is basement membrane, and in the kidneys, in the glomerulus, there is basement membrane. Here there is basement membrane, here there is basement membrane. This basement membrane is made up of some type of collagen, and particularly here there is type 4 collagen. Okay, here there is in the basement membranes of lungs and in kidneys. In the kidneys means in the glomerulus, there is type 4 collagen. The antibodies are produced against this type 4 collagen. The antibodies are produced against this type 4 collagen. So for example, if we take in kidney, what happens? The kidney glomerular basement membrane is what? Due to lack of collagen, due to destroying of collagen by antibodies, what happened? This basement membrane gets what? See, if membrane is like this, strong, and somewhat it destroyed, it became weak. So large molecules which should not be filtered into the urine will filter. For example, proteins will go. For example, proteins will also leak from it. Other substances also like glucose, those also will go. But for most prominent things we should see, those are first proteins are going. Proteins are leaking out. Okay, blood also going out. So we can see hematuria. Blood is going out, proteins also going out. So we can see proteinuria. Okay, see, we can see proteinuria, we can see hematuria. Why? Because the basement membrane is destroyed. And in the lungs, due to destroying of basement membrane in the lungs, the blood will come out into the lungs. Into the lungs it come out. So whenever the person coughs, there will be hemoptysis. Okay, what happened in the Goodpasture syndrome? It will mostly affect the type 4 collagen which is there in the basement membranes of the kidney and the lungs. So there will be destroying of the glomerulus and also the lungs. So in the lungs there will be hematemesis due to leaking of blood into the lungs. And in the kidney there will be loss of protein, there will be loss of blood. So we can see hematuria and proteinuria. Okay, this is the classical triad. This is the classical triad of Goodpasture syndrome. What? Classical triad, that is hematuria, hematemesis or hemoptysis and proteinuria. Okay, this is the classical syndrome of type 2 hypersensitivity. So we nailed everything out